Good evening, namaste. Welcome again. We have completed 6 months of uninterrupted integrative health education series. And this is a part of biology series in which we're trying to arouse your interest and tell you simple things, basics about various aspects of biology. We had some discussion on DNA, RNA, and we will have a follow-up discussion on transcription, translation. Today is a very interesting day because a lot of you have been hearing about stem cells, and these are cells, but how specialized they are. Why do they arouse so much interest and interest of ethics and ethics committees, applications and false claims and good things coming out? So we thought we will combine two people who have worked at different knowledge levels. One is Dr. Saras Jyoti. She's a postdoctoral fellow at the Haryana Yoga Council, and Dr. Peter Andrews. The entire session will be coordinated by Dr. Parul Bali, who's a postdoctoral fellow at the PGI Chandigarh. And I hope you have a great session, and let us also let's ensure that this becomes the bedrock for future studies and collaboration and more research into alternative systems as well as the mainstream modern medicine. Thank you very much for your attention. Over to Dr. Parul Bali.
Good evening, everyone. Now is the sixth session of our biological series. Today's topic is on basic principle of stem cell biology and their application by our speaker, Dr. Saras Jyoti. She has done Masters in Stem Cell and Tissue Engineering in 2010 from Punjab University, Chandigarh. She completed her PhD from Department of Biotechnology and Informatics from JP University of Information and Technology from Himachal Pradesh. She worked as a research associate in Amity Institute of Molecular Medicine and Stem Cell Research, Amity University, Noida, India. Along with Dr. Saras, we have a very special guest as a commentator for today's presentation, Dr. Peter Andrews. He has done bachelor's in biochemistry from University of Leeds in 1971, DPhil in genetics from the University of Oxford in 1975. Following postdoctoral research at the Institute of Pasteur in Paris and Sloan Kettering Institute in New York. He was a research scientist at Wistar Institute of Anatomy and Biology. While in Philadelphia, he has also obtained MBA degree. At the Wistar Institute, he characterized the properties of human embryonal carcinoma cells, which are the malignant counterpart of human embryonic stem cells, and identified surface marker antigens now extensively used for monitoring human embryonic stem cells. He also characterized a pluripotent human embryonic cell line, NTERA2, which is widely used in studies for human neurogenesis. In 1992, Peter Andrews was appointed to the Arthur Jackson Chair of Biomedical Research in the University of Sheffield. His research continues to focus on the biology of human pluripotent stem cells and particularly their mechanisms of weight determination, notably to neural crest derivatives, as well as their susceptibility to genetic change upon long-term culture. So with this introduction, I am pleased to begin our session and invite Dr. Saras Jyoti to present. Over to you, Saras.
Thank you, Parul, for the introduction. Is it okay?
Yes, it is visible. We can start.
Yes. Good evening, everybody. I welcome you all. Myself, Dr. Saras Jyoti, and I'm going to present a talk on the basic principle of stem cell biology and their application. Now, starting with the history, in early 1960s, Till and McCulloch, while studying the effect of radiation on bone marrow, they demonstrated the existence of multipotent stem cells, that is hematopoietic stem cells. Later, Altman and Das presented scientific evidence of neurogenesis, but their reports were largely ignored. In 1978, hematopoietic stem cell in human cord blood cells were discovered. And in 1981, mouse embryonic stem cells were derived from the inner cell mass by scientists Martin Evans, Matthew Kaufman, and Gail R. Martin. And further, in 1996, the famous cloning of dolly shape was discovered by somatic cell nuclear transfer. And later, in 1997, Bonnet and Dick showed for the first time that the leukemia originated from a hematopoietic stem cells, which is the direct evidence for cancer stem cells. And later in 1998, James Thompson and co-worker derived the first human embryonic stem cell line. In 2000, there were several reports published for the adult stem cell plasticity. And in August 2006, very interesting reprogramming of differentiated cells into undifferentiated cells were discovered by Takashaki and Yamanaka, which were known as in mouse-induced pluripotent stem cells. Now, what is a stem cell? Stem cell is a cell that has the capability to self-renew and have the potential to differentiate into various cell types in the body. Now, what are their characteristics? They're unspecialized. They have capability of self-renewing themselves for a long period of time. They have the potential to give rise to specialized cell types, the process known as differentiation. Now, they are classified into three broad categories based on their differentiation potential. That is totipotency, pluripotency, and multipotency. Now, what is in this diagram? We will learn, this diagram will eventually explain all the process starting from the fertilized egg to a mature cell types. Here we can see these cells which are in red color, the 8 cell stage are known as totipotent stem cells. They can give rise to the embryo as well as placenta. Later, the stages, there is a formation of a blastocyst, and in this blastocyst, there are cells in the purple. We can see they are pluripotent nature, and these cells can just form the embryo, but not the extraembryonic tissues. And further differentiation takes place. The cells become multipotent and further into mature cell types. Now, there are two types of stem cells: embryonic stem cells and adult stem cells. Embryonic stem cells come from a 5 to 6 day old embryo, and they have the ability to form virtually any type of cells found in the human body. And adult stem cells, they are undifferentiated cells found among the specialized cells inside the tissue and organ, but they have a limited potential to produce other cell types and self-renew. Now, stages of embryogenesis. We have to understand the stages of embryogenesis to understand the isolation or activation of isolation of both the cell types. Now, when fertilized egg gives 2 cell staged, at day 3 and 4, there are multiple cell types. And at this stage, we isolate the cells from 5 to 6 days blastocyst, from the inner cell mass of the blastocyst. And day 11, it is a day of tissue differentiation. Now next, how to derive cell from the inner cell mass. These are the outer layer of cells which forms the placenta, and inner is the inner cell mass which forms the fetus. Now, this process destroys the embryo, which is an ethical issue we'll discuss later. But here we isolate embryonic stem cell from the inner cell mass and culture them and differentiate them into various cell types depending upon the requirement. But the special factors which can differentiate into other cell types, they require different growth factors, which are still largely unknown. Now, characterization. For a cell line to be for any cell, there are some important criteria which need to be satisfied before a cell line to be considered as a bona fide embryonic stem cell line. These are the first, they should express the cell surface marker. They should have pluripotency factors, OX, NANOG. They should express high telomerase activity They should have normal karyotype. They should have the potential to form embryonic body in vitro, and they should form terotoma formation into mice containing all germ layers. And pluripotency in vivo, that is known as chimera formation. Now, why these embryonic stem cells are so valuable? Based on their potential to give rise to 200 cell types, these cells can be differentiated into many cell types when given correct differentiation growth factors, and can be used for any degenerative disease present at the stage. Now, coming to adult stem cells. Adult stem cells reside inside our body. They reside in a special place called stem cell niche. They contribute to the homeostasis. They divide only when needed. Their progeny differentiate into cells that perform essential body functions. Now, where these adult stems can be found, they are many unidentified or identified in many organs and tissue. They are very small in numbers, so it is very difficult to locate them, isolate them practically. So, some stem cells are thought to reside in specific area tissue where they remain quiescent until they are activated by an injury. Or the most studied adult stem cells are bone marrow, brain, and liver, kidney, and skin. Now, the cultivation of stem cells. The stem cell has 4 stages of differentiation. First, you isolate the stem cells, then you characterize them, then the cell maintain the stable cell line for a long period of passaging, and then you can control and action the stem cell differentiation into desired lineage, which develop into functionally active cells. Now, what are the advantages of embryonic stem cells and adult stem cells? Embryonic stem cells, they are pluripotent in nature, can give rise to more than 200 types of cells. They are stable and can go many divisions. They are easy to obtain, but their embryo destruction is an ethical issue. There is a possibility of immune rejection. They have high potential for tumor formation. Now, on the other side, adult stem cells are multipotent. They have restricted differentiation ability. They are less stable. Capability is less for self-renewability. No ethical concern. That is good. But they are difficult to isolate in the adult tissue. And host rejection is minimized in adult stem cells. And they have less tumorigenic potential as compared to embryonic stem cells. Now, the barriers of these ESL-based therapies, like they have legal and political issues, they have immunological incapability, there are safety concerns of tumorigenicity, and the alternative sources of pluripotent stem cell is the induced pluripotent stem cells, which were discovered in 2006, as I explained in the second slide. The skin cells, they were specialized, they were injected with the cocktail of auxin, CMYC, and KLF4, and they differentiated, they would become unspecialized from specialized cells, and they have the ability to self-renew and differentiation. Now, these cells, which are induced pluripotent stem cells, they have the potential to become patient-specific or disease-specific, and then have various potential in the field of developmental biology, disease mechanisms, drug development, cell replacement therapy. Now, derivation of human iPS, the efficiency of reprogramming ranges from 0.02% to 0.002%. That is very, very less. So thankfully, but we have these systems. Induction of pluripotent stem cells from adult human fibroblasts by defined factors is done by Yamanaka et al. And induced pluripotent stem cell derived from human somatic cells is done by Thompson J. and team. Reprogramming of human embryonic stem cell to pluripotency with defined factors was published in Nature 2008. And generation of human-induced pluripotent stem cells from dermofibroblasts were again published in 2008. Now these induced pluripotent stem cells have the various application. They can be used in cell therapies, drug target validation. So they are, now we have 3D print ink, bioink systems. They can be used as drug delivery. They are used. They have been used in toxicology screening and obviously as a research tool. Now, based on cell replacement therapy, cell could be stimulated to develop into specialized cells that represent renewable sources of cells for tissue for transplantation. This cell therapy could treat injuries, various genetical disorder, degenerative conditions like Alzheimer, Parkinson's, arthritis, diabetes, spinal cord injuries, blood disorders such as hemophilia. Now, as I discussed earlier in the slide, any disease which is where there is a tissue degeneration can be potential candidate for stem cell therapies. Again, the other potential is development and testing of the drugs. Researchers could beneficially test in the less time, at less cost, the toxic effects of medicines on pluripotent stem cells that have been developed to make the disease process. Now, again, there is an advancement in the research of stem cells. Researchers are using bioink printers to develop organoid structures. Now there is and stem cells can be used as for understanding the developmental biology. The formation of 3 germ layers from the inner cell mass can lead us to understanding of the various differentiation potential of various organs. So we understanding the developmental biology will guide us for the regeneration potential of embryonic stems. And there are major progress in several important health areas such as Alzheimer's disease, Parkinson's, spinal cord, heart, severe burns, diabetes. And there are more than 5,000 registered clinical trials involving stem cell research. In India, as well as globally, only blood stem cells from bone marrow to treat blood cancers and different blood disorders are permitted. And the clinical use in other disease or any, it's still in infancy. At present, for which diseases stem cell therapies is available. At present, we have only bone marrow transplant or hematopoietic transplant. For other diseases, the research is still being conducted to see if stem cells can be helpful in curing these diseases. These are some pictures showing just the status of some clinical trials which has been registered. And looking at the current scenario, stem cell therapy and COVID-19. Majority of clinical trials include mesenchymal stem cells application. The mesenchymal stem cells with the people who have patient who have COVID-19 and viral pneumonia, it was found that the mesenchymal stem cell were able to maintain the health of these patients who are suffering from viral pneumonia. These are the clinical trials which have been published recently. Now, this challenges to stem cell research. The cells won't be easily necessary to, when injected inside the body, they will not develop into proper functional and mature cell type. They may be instead form a tumor. We need to control the differentiation first to provide the regenerative medicine. The obstacles of stem cell research: how to find the right type of stem cell, how to put the stem cell into the right place. Will the stem cell perform the desired function in the body? Or differentiation protocols for many types have not been developed till date. With this, I hope you understand, and thank you.
Thank you, Dr. Saras, for your presentation. Now I invite Dr. Andrews to give his presentation and your valuable comments. Sir, kindly share your PPT.
Okay. Well, thank you very much. That was a really very good summary of the stem cell field, and I hope everyone who's not familiar with the area will have gained some insights into what is possible and also some of the difficulties. It's clearly what has excited everyone, as Dr. Jyoti said, is the possibility of using stem cells to create cells that could be used to replace diseased or damaged tissues. And she mentioned a number of studies that are in progress or soon expected to be in progress. For example, using pluripotent stem cells to create retinal pigment cells for transplantation to try to recover sight or prevent loss of sight in people suffering from age-related macular degeneration. And there's a number of clinical trials of that are in progress. And Parkinson's is another area where there's a lot of excitement because there's evidence that fetal neurons can be used to transplant to Parkinson's patients and affect a cure. But of course, obtaining fetal neurons is very difficult in a very standardized way. And there's a lot of people working to derive dopaminergic neurons from pluripotent stem cells in a way that could be very easily standardized and so the cells can be used repeatedly. And there's a number of trials anticipated in that direction. In fact, I believe there's already a trial started in Kyoto in Japan. So it's a very exciting area, but also, as Dr. Jyoti mentioned, we can use these cells for other things, not just replacement therapies, but potentially for exploring disease mechanisms, for exploring problems in embryonic development, and also in using the cells to screen for screening potential chemicals for drugs. And these are areas that many people are pursuing. I thought one thing I would just touch on, as Dr. Jyoti mentioned, one of the potential problems with pluripotent stem cells is the issue of tumorigenicity. And I thought I would just expand a little bit on this, if I can share my screen here. So actually, one of the things with pluripotent stem cells is that when you if you just take pluripotent stem cell and were to place it in an immunosuppressed mouse, for example, it will form a tumor. And the tumor is typically, as Dr. Jyoti mentioned, a teratoma. And that is a teratoma, and here's an example. Let's see if you can see this. This is a cross-section of a teratoma in which there's a lot of different sorts of tissues. And a teratoma is essentially a tumor that contains somatic tissues of all three germ layers. So here, in fact, there's some endoderm tissues here. There's some neural tissue here. This looks like cartilage down here. All sorts of different tissues. It's as if you've taken an embryo and jumbled it up and got all the bits there together. Now, on the whole, those tissues are probably benign and not tumorigenic, but the problem is that these tumors may contain persisting undifferentiated cells, and there's a picture of some here. These are undifferentiated embryonic stem cells, and they're very similar to what are called embryonal carcinoma cells that one finds in real tumor and germ cell tumors, which are very similar kind of tumors that occur in people. And those arise from germ cells. And these cells, the embryonal carcinoma cells, are very tumorigenic. So what you typically have is you can have a tumor which contains differentiated cells, and those are known as teratomas. And with some caveats, they're probably not likely to form tumors. They may do, but very rarely. But if they retain these population of undifferentiated cells, and sometimes when you inject these cells into mice, or we imagine if you put them into people, obviously you wouldn't do that, they could form what is called a teratocarcinoma, which is a teratoma which also contains a population of pluripotent stem cells. Now, there's been a lot of concern about tumorigenicity from embryonic stem cells, but it's really focused on the potential for pluripotent stem cells to persist and form a tumor. But of course, when we're doing regenerative medicine, you would not really want to implant these cells. What you would like to do is differentiate the cells into something, progenitor cells for, say, neurons, or progenitor cells for liver or whatever. And those would be the cells that you would transplant into patients. And it's reasonably to expect that these cells are probably not, or only going to be very rarely tumorigenic. So the key thing to avoid tumorigenicity with these types of treatments is to make sure that in preparing cells for transplantation, that we don't have any of these pluripotent stem cells persisting. However, there is a caveat, because clearly these cells could develop into tumor cell in their own right. And that is what is a concern, is whether these could develop into somatic tumors. So they probably would do it rarely. But another aspect of pluripotent stem cells, which is relevant to this, is that we know over a period of time, if we culture them in, for a while, they acquire mutations. And some of these mutations give rise to variant cells which have a growth advantage. And if they have a growth advantage and you grow these cells in culture, they'll eventually take over the whole culture. So it is possible that you end up with cultures of pluripotent stem cells that are not karyotypically normal, as Dr. Jyoti said, is the pristine state, but may have a genetic change. And we have been studying those a lot. They could involve small nucleoside changes, or they could involve whole chromosome changes. For example, gains of whole chromosomes or gains of bits of chromosomes. And it turns out, when we grow embryonic stem cells, is that there's some chromosomes which are much more likely to crop up as variants in pluripotent stem cells over any length of time. Typically, this is an example from the literature. Each of these are the different chromosomes in the human karyotype, and the bars represent examples where a cell line has acquired an extra copy of the bit of chromosome that the bar is against. Green means they've gained that bit of chromosome, red means they've lost. And as you can see, some chromosomes are almost never changed, like chromosome 4, whereas chromosome 12, 17, 1, and 20 are frequently affected by gains of pieces of chromosome. And we think that that's because there are genes in there that give the cells a growth advantage if you have extra copies of them. So a concern with that is, could those sort of variants also then feed back and give you problems? Could those include variants which could turn some of these cells into tumorigenic cells? And that is certainly an area that we're investigating and trying to understand something about the relationship of genetic stability of these cells, and also the potential for some of these cells to develop as tumors. So, as I said, these are concerns that are being investigated at the minute, but they're probably not terrible. In one of the big studies that we did as an international consortium, looking at something like 120 different cell lines, actually 79 of them over a period of time stayed genetically normal during the study. So it's a concern, but it's not a concern that we should be overly worried about, in the sense that we shut up and go home. It's something that we think that we can manage. So I think I'll just leave it there. That, as I said, Dr. Jyoti gave a very good summary of the area. I thought I'd just add that a little bit in about tumorigenicity. And I think now we perhaps turn this over to questions from the audience.
Yes, sir, I request you to unshare your slide.
Yes. Stop sharing. Yes. Okay. There we go.
So now we'll take a few questions, and before that, I want to ask you something. So apart stem cell for therapeutic purpose, they are being used as a cellular model or for studying developmental of the some cell type. So if we are using as a cellular model, there are of two types. One is the patient-derived iPSC based cell lines, and another are the cell lines, differentiated cell lines, which are then produced by creating some mutation and then used as a cellular model for testing some drug. Out of these two models, which one are the best to use and to study?
Would you like me to address that, maybe Dr. Jyoti? I'll answer first, and then actually Dr. Jyoti might want to add some things. I think the answer is there is no best model. I think you have to consider things for the specific question you're trying to ask. You have to remember, one has to be slightly careful with iPS cells, is induced pluripotent stem cells. These had been somatic cells. We've reprogrammed them back to a pluripotent stem cell state, like an embryonic stem cell state. So in an ideal circumstance, an iPS cell and an ES cell are exactly the same thing. In practice, there's the possibility that one hasn't completely reprogrammed some genes, that there may be some differences. You have to be a little bit careful, particularly with regard to the epigenetics of this. But I think it depends what your questions would be that you're trying to answer. If you're looking, your access to iPS and ES cells may be to obtain ones with carrying mutations that affect particular pathways that you think might be important in particular developmental processes. There's obviously no reason why you can't introduce particular mutations that might be of interest to you. In terms of you mentioned drug discovery. I mean, one of the areas that has been of particular interest is in the cardiac field, where people have been looking at the response of drugs that might cause things like long QT syndrome and sudden cardiac arrest in some patients. And people have been looking at mutants, using mutant variants of cardiac cells derived from embryonic stem cells to test whether different drugs might influence those aspects of cardiac cell physiology, for example. So the quick answer is that there isn't a best cell type. I think one has to address it in the context of what your particular question is.
Okay, sir, I want to know, is there any major difference between mouse embryonic stem cells and human embryonic stem cells?
So, well, that is also an interesting question. There clearly is, in terms of, if you look at the general thought at the moment is that mouse pluripotent stem cells correspond to a pre-implantation primitive ectodermal stage, the sort of late primitive ectoderm of the inner cell mass. The human cells appear to correspond to a slightly later stage, the peri-implantation stage, and they've been referred to as naive pluripotent stem cells, as opposed to, as prime pluripotent stem cells, as opposed to naive pluripotent stem cells, which is what mouse pluripotent stem cells or embryonic stem cells have been christened. And indeed, from the mouse, you can obtain cells from the peri-implantation stage, which are referred to as epiblast stem cells. And they seem to be much more similar in some respects to the human cells. So one notion is that so-called human embryonic stem cells are really epiblast stem cells, as opposed to the mouse embryonic stem cells, which correspond to an earlier stage. And people have derived, by various manipulations, human cells which more closely resemble the mouse. However, it's not nearly as simple as that, because the human ES cells come from human blastocysts pretty much at the same stage that the mouse embryonic stem cells come. So the question is, why do you get something that appears to be slightly different? And indeed, the human cells are not, although they're similar to epiblast stem cells from the mouse, they're not the same. So one of the problems we have is that in trying to understand the biology of human embryonic stem cells, we always refer back to mouse embryos. And it's clear that mouse and human embryos do develop somewhat differently. And so what is one of the areas which is important, and there's a number of groups around the world focusing on it, is looking at the early embryology of human embryos so that we can relate the human embryonic stem cells to real human embryos as opposed to trying to guess what they might be because we have to relate them back to the mouse. So the quick answer is mouse and human embryonic stem cells are slightly different. The reasons for that are not entirely clear. Is it because they represent a different stage of development, or is it because humans are not mice? And I think the jury is out on that score. It's probably a bit of both.
Okay, thank you, sir. One last question from my side.
Yes.
The issue of carcinogenesis, there is a property of carcinogenesis with the pluripotent stem cell. So such properties are also with the mesenchymal stem cell, because most of the research says that these are very safe cells and they have some immunomodulatory activity. People are injected into the blood, which is very safe. So is there any possibility that these cells can go out of control and give some carcinogenic property?
Which mesenchymal stem cells?
Yes.
I have to say I don't really know a huge amount about them. One of the problems with so-called MSCs, mesodermal stem cells, is that they're really actually very ill-defined. And a lot of what people are calling stem cells are really not stem cells, and they're often mixed populations of cells. So they're really not very well-defined cells in the first instance. Of course, it is certainly possible that they could generate tumours, but I don't really know what the frequency of that would be. And it's not clear that it would be any different for the differentiated derivatives of pluripotent stem cells. As I said, the real concern, most of the concern about tumorigenicity from pluripotent stem cells has revolved around the tumorigenicity of the undifferentiated cells, and those are the cells that you would not want to transplant into a person in any case. So the tumorigenicity of the differentiated derivatives may well be much, much less and comparable to any other somatic cell.
Thank you, sir. So anyone have any question?
Yes, good afternoon, good evening, sir. Sir, I want to ask that does the health of a person have any effect on the potency of stem cells? That if we isolate stem cells from a healthy person and a person with a comorbidity, does these stem cells have any difference in its potential or its in regenerative capacity or not?
Well, I think it depends. So it depends, first of all, which sort of stem cells you're talking about. Are we talking about embryonic stem cells or?
Sir, I want to ask about umbilical cord blood stem cells.
Okay. I mean, the quick answer is, the quick answer is I don't know for sure, but you would expect that it would depend on what the condition of the patient is. If you're talking about umbilical cord blood cells, they would presumably relate to the state of the fetus or the baby from which they've come. And there may well be some conditions that would cause them problems and other conditions that they wouldn't be. So I don't think there's a very simple answer to that question. I think you've got to know the specific problem and then think it through and say, well, is this particular disease condition going to give me a problem with is likely to cause a problem with those cells? So I don't think we know in a general sense. I think you've got to focus the question on particular conditions and then address whether that may or may not influence the behavior of those cells in that circumstance.
Is there any question from anyone?
Hello.
Hello?
You are audible.
Yes, ma'am. I want to ask that umbilical cord blood cell, derived stem cells, and mesenchymal stem cells come under which category? Do they come under embryonic stem cells category, or they are separate from both the category, adult stem cells as well as embryonic stem
They
cells?
are separate categories. They don't come under embryonic stem cells. Umbilical cord stem cells are different, and mesenchymal stem cells come under the adult stem cells.
Okay.
I think actually it's sometimes misleading to refer to these as adult stem cells. What we're really talking about is somatic cells. They're cells that so-called adult stem cells could occur in the fetus, but they're quite different from what we're calling embryonic stem cells. So they're cells related to specific tissues, and they're involved in replacing and developing those particular tissues. So they have very specific limited capacities for differentiation, which is very different from embryonic stem cells.
Okay. Thank you, sir.
Yeah, just a moment.
Is there any question from anyone?
Please go ahead.
So I'll ask a question to Saras.
Yes, sir.
Saras said you showed in your slide that there are a lot of applications of stem
Yes, sir.
cells in Parkinson's, Alzheimer, diabetes, but so far we haven't seen any good outcomes. Or do you have information of a very good outcome, or in the coming years that you put it in your slide?
Sir, there are many progresses made so far. In their clinical trials, which has been successful for retinal regeneration, and with mesenchymal stem cells, there are many clinical trials ongoing for regeneration of. I cannot particular say for any particular reason, but there are even with the COVID-19 patient, they have seen that mesenchymal stem cells when they injected mesenchymal stem cells in the COVID-19 patient who are also suffering with viral pneumonia, their health condition was improved. And out of I don't know the exact number, but out of 4 or 5 patients were healthy after the treatment with the mesenchymal stem cells.
So let's ask the expert, Dr. Andrews.
Yes, sir.
What do you think, Dr. Andrews, is the next?
So I can really only comment on pluripotent stem cells, and I think I think the important thing to make sure everyone's aware of is that these are very, very, very early days, and one of the big concerns of everyone in the field is that there's a lot of somewhat unscrupulous people around the world who are trying to sell stem cell treatments which are completely unvalidated and untested just because they can get money out of people. So I think that's one very important thing to say. There's been clearly some, very limited success so far with retinal pigment cell transplantation, but again, it's very early days. My colleague Pete Coffey in London, I know, has transplanted 2 patients, and let's say the results were promising. And so far, no problems. Parkinson's, I think, is the next big area that people will be trying to develop. And I know there's a number of groups around the world focusing on transplantations in that area. So John Takahashi in Kyoto, I believe, has already transplanted a patient or some patients. I know that Lawrence Studer group in the States is interested in is sort of nearly ready to do some sort of transplantation. My colleague in England, Roger Barker, who's working with Malin Palmer in Sweden, they're in a position, hopefully in the next 2 or 3 years, to be transplanting into Parkinson's patients. I think I'm not that familiar with the cardiac field, but there's certainly some you know, at least on the horizon, there's some possibility of doing some transplantation for hearts. And of course, there's been some spinal cord work. So I think the important thing just for everyone to keep in mind is it's really very early days yet, but there's a number of promising conditions. Of course, diabetes is the other obvious one.
Wherever the target is more defined and more specific, maybe there is more chance.
Yes, I think that's a really important point. Where you have a very specific cell type that is lost and hopefully doesn't depend too much on a particular structure, you have more chance of success. The other is the issue of numbers of cells that you've got to transplant and things like the age of the patient. And I think an issue to consider, for example, if we take age-related macular degeneration, dealing with a condition affecting old people on the whole, people who their sight is going to be lost if the condition continues. There's no available treatment now, at least for dry AMD. You need a very small number of cells. You're putting them into a defined organ, the eye, and you can monitor it very easily. So that's one scenario. The other scenario would be, say, diabetes. And if you were trying to transplant beta cells into people, you'd be dealing potentially with much younger people. You'd have to be put a large number of cells in. They're people for whom there is already existing treatment. Now they can take insulin injections. For the most part, that works. And so the risk assessment, I think, has to be very different. Whereas you may be not too worried or less concerned about untoward effects in treating AMD, you'd be much more concerned in the case of treating diabetes in young people. So these are sort of things that we'll have to consider as well.
Do you think the damage, the severity of the damage or disease may be another factor? What do we know from literature?
Well, I mean, I think that's certainly the case as well. Certainly in the case of AMD, the reason for transplanting retinal pigment cells is to support the photoreceptors. But of course, if the disease has progressed to the point that the photoreceptors in the macula have all degenerated, then you can't replace them. Parkinson's, I don't know, but of course, again, it's not entirely clear how these cells will really work. Will they really integrate, or are they just going to be cells that are sort of pumping out dopamine in the right spot? I think the answer is we don't really know, but these are certainly conditions to think about. The other, of course, in the case of diabetes, there's always the concern that this is an autoimmune disease potentially, and that if you haven't dealt with that, then the transplanted cells may actually be targeted by the immune system anyway. So there may be problems. So I think there are a lot of problem issues along these lines which have to be considered.
Most of our,
immediate answer.
Sure. A lot of our viewers are always interested to know, as they hear more and more about banking, cord banking and bone marrow banking, what is your advice and sort of how should they address this? Is this just a hype, or maybe it's not that bad?
Personally, I think it. So this all started some years back when there was a lot of argument, or a number of papers and people suggested that so-called adult stem cells, particularly hemopoietic stem cells, might be able to be induced to differentiate into all sorts of other tissues. And I don't think people accept that. That doesn't seem to, in fact, be the case. And I found that there were, I found it slightly unscrupulous people pushing the idea of banking cord blood because you might be able to cure someone in the future with all sorts of diseases that that might be curable using those core, and there's really little evidence that that will be the case. I mean, even in the case of hemopoietic, the one condition where things are used clinically is transplanting hemopoietic stem cells. And my understanding is that actually and the idea will be with cord blood, you could have your own stem cell. But my understanding is that actually the number of stem cells in cord blood is not sufficient to actually transplant and repopulate an adult patient. Now, that might be changed in the future if people can get the cord blood hemopoietic stem cells to proliferate, but I don't think that's.
So why do you say that? It is a lot of, it's abundant cord blood cells.
Yeah, but there's not enough. Actually, I think if I understand the amount, if you wanted to replace hemopoietic cells in an adult, you need sort of 3 or 4 lots of cord blood.
Correct.
To give you enough. That's my understanding. Yeah, I have to say it's not my area, so I'm not a hematologist. But my understanding was there may be enough to transplant into a child, but not enough into an adult.
Sure. Are any of the yoga scholars, Parul, you want to ask if they want to? Any of the yoga scholars, just any question, very naive, or you have a stem cell expert and people are listening? Or else we can request Dr. Andrews to give a kind of a direction where the next thesis, PhD thesis should work on, or any new research. What is the way forward from having been involved in discovery, even when the hype came? I recall you've been involved in the discovery of the embryonic stem cell markers and most fundamental work, having gone this 40 years of research, 50 years of, what is the way forward?
I think one of the key areas going forward is figuring out how to induce these cells to differentiate into the cells that you really want. And one of the things just to keep in mind, of course, is that a lot of the differentiation protocols today are very crude. They sort of think, okay, we throw this growth factor on for 2 days and we add this and we'll add this. It's kind of cookery. And the reality is that in the embryo, people are trying to recreate what goes on in the embryo. But of course, in the embryo, cells are not exposed to, I don't know, BMP for 3 days and FGF for 2 days. They're exposed to pulses of this under very specific conditions, under particular times, for certain periods of time, in specific combinations. And the sort of whole bucket level approach is really very crude, and clearly it works, and we're having some success in getting certain cells out. But I think there's a lot of opportunity to make that a lot more refined over the next few years, and figuring out how to make cells differentiate and how to get the right sort of cell out. I mean, even in the Parkinson's field, in work from Malin Palmer's group, for example, first of all, they were clearly finding dopaminergic neurons related to the substantia nigra that they could get from embryonic stem cells. But then actually subsequently, they discovered that there were two different groups of cells they were producing. And it was only one of those groups which was suitable for getting, at least in the animal models, reconstitution of functional dopaminergic production in the brain. So again, there's just one little example, but I think this is one of the big areas will be how to refine the differentiation of the cells to get exactly the cell you want, and how to recognize what that cell is. Clearly that there are other problems. My own area is this issue of genetic stability, and we don't actually think that these cells are particularly unstable. They do acquire mutants because of selection during culture and maybe during differentiation that may be ones you don't want to have in your end product. So understanding those will be equally important.
Yes.
So there's a big area, and I think there's plenty for people to keep themselves busy for the next 50 years.
Sure. Thank you,
Sir,
Dr. Andrews.
I wanted to ask a question that can stem cell therapy help in hypoxia conditions as well, or is there any studies or about that?
So I didn't catch that.
Can stem cell therapy help in hypoxia conditions as well? Hypoxia conditions.
In hypoxia,
Yes, sir.
I don't know. I mean, actually, one of the issues with hypoxia is that we do know that it looks like that the cells are less genetically more genetically stable if you grow them under low oxygen conditions. That's one aspect. Maybe, although it's actually quite difficult to do it properly.
A lot of people actually, Dr. Andrews, a lot of investigations over around clinical issues and disease and humans. Of course, a lot of people like you or the basic scientists take things down to cell culture level and look at the biology and induction and hypoxia and different environments, damage them and rescue that. But we work in a medical institute, and most often the physicians would like to know the applications, and they would like to know if stem cells are good markers of disease or health or acclimatization. I think this girl,
I
Sheetal,
see.
yeah, that's the idea she's throwing at you.
I think this is an area where it's important to actually have close collaboration between basic scientists and clinicians, and have kind of clinicians who have an understanding of basic science, and vice versa. So I think this is a whole area where it's important to have clear collaboration with people with different areas of expertise, and I think that's probably another thing to kind of keep in mind in developing this field forward.
Yeah, but what Sheetal, that girl, was trying to ask is if you take some group of individuals to a high altitude with hypoxic conditions and with the challenge to acclimatize faster, will stem cell play a role, if at all, they are in the scheme of things of acclimatization or adaptation, or does the hypoxia just bump up the stem cell reservoir?
I honestly don't know. It's actually rather outside my field.
Yeah, sure.
It would be an interesting question to address, but I don't know if anyone has done that.
Yeah, I'll let Parul take over and take the proceedings forward.
Okay.
Dr. Andrews and Dr. Saras for a wonderful presentation. Thank you, sir, for your kind and valuable suggestions. Thank you very much.